Okay, just 3 more slides to go. Another thing we like to do is to combine antibiotics. There's several reasons why, and some of the results that it can produce is, one, what we call synergism, and this is when the 2 drugs together have an effect that is greater than either of them could have on their own. It's kind of like 2 plus 2 doesn't equal 4, 2 plus 2 is going to equal 10. Example of this is that penicillin and streptomycin are more effective when they're taken together to combat bacterial endocarditis. Some benefits of using the 2 drugs together is that it can prevent drug resistance. If a bacteria develops resistance to one drug, if another drug is present, it can kill that resistant strain before it can proliferate and give rise to a new resistant strain. It can also allow treatment to be provided prior to diagnosis. If a doctor doesn't know exactly what pathogen is causing the disease, then he or she can give more than one antibiotic that will, one of them most likely help cause, sorry, this is so awkward when I'm not in front of a class. When they're used together, one of them at least one is likely to kill the bacteria if the other one doesn't. The other thing that using 2 together can do is lessen the toxicity of individual drugs. Using some antibiotics in the presence of another antibiotic will actually reduce those toxic effects. However, using 2 drugs together can result in antagonism, which is when you have 2 plus 2 equals 1 or 0. We see this with penicillin and tetracycline. If they're used together, they are much less effective. So as resistance increases among bacteria, we have to look for more ways to kill pathogens. Some new methods that we have come up with include using antimicrobial peptides and antisense agents. Antimicrobial peptides are produced in many higher organisms as defense against microbes. They can disrupt microbial membranes and enveloped viruses, or they can disrupt attachment of non-enveloped viruses. These antimicrobial peptides can be found in a number of different organisms, including frogs and moths. Niacin is found in lactic acid bacteria, and it's an AMP or antimicrobial peptide that has been used in food preservations for decades. However, we've not seen any new resistance. We've also recently discovered that there are many types of antimicrobial peptides that are very effective and could also thwart the development of antibiotic resistance. Research is being done on shark steroids to boost the immune system. Titin, I don't know how to say it, is a small molecule that blocks virulence factors in bacteria. So there's just a lot of different things that we can start looking at instead of just antibiotics. Phage therapy I'll actually talk about on the next slide. Antisense agents are short synthetic DNA strands that can selectively recognize and bind to specific target sites on pathogenic DNA or RNA. And when they bind to those sites, it will block biosynthesis of essential proteins, and therefore the pathogen cannot replicate, cannot metabolize, can't grow. One example of this mechanism is that mammalian cells have a mechanism that can prevent RNA from giving rise to the proteins that they encode. It'll just interfere with the RNA synthesis and the whole process that brings about proteins. This mechanism is called RNA interference. It is abbreviated RNA and then a little I. It's the basis of drugs that are called small interfering RNAs, and these are abbreviated siRNA, and these can selectively block protein synthesis in pathogens. Phage therapy uses highly selective bacteriophages to attack bacteria. Remember, a bacteriophage is a virus that only infects bacterial cells. This seems to have a whole lot of possibilities to it, however, it is still under study, so hopefully we can see more of that in the future. And that is the end of Lecture 20. I hope you guys have had a really great, fantastic, wonderful time listening to this on your 4th of July weekend. I'm sure it is everything that you wanted to do. So I guess unfortunately now you'll have to get back to enjoying yourself. Have a great weekend, and I will see you all on Monday.